Blood is not identical in every person. Blood type describes inherited markers found mainly on red blood cells. In this lesson, we will examine blood's components, the ABO and RH systems, and the principles of transfusion compatibility. Blood type is inherited from biological parents through genes. The ABO system provides 4 groups: A, B, AB, and O. Adding the RHD marker creates the 8 common types, each labeled positive or negative. Blood contains 4 main components. Red blood cells transport oxygen. White blood cells support immune defense. Plasma is the yellowish liquid carrying proteins and salts. Platelets help the blood clot after an injury. Red blood cells have a flexible biconcave shape. Inside them, hemoglobin binds oxygen in the lungs. The cells then travel through blood vessels and release that oxygen to tissues throughout the body. White blood cells help identify and respond to infection. Plasma carries cells and dissolved substances through the circulation. When a vessel is damaged, platelets gather at the site and help form a clot. On the surface of each red blood cell are molecular markers called antigens. Blood group antigens may be proteins or sugars. Small inherited differences in these markers help determine a person's blood type. The molecules carrying blood group antigens may also transport substances or support the red cell membrane. For blood typing, their key role is identity. The immune system can recognize familiar markers and react to unfamiliar ones. Antibodies are defensive proteins found in plasma. They can bind to antigens that the immune system considers foreign. In the ABO system, anti-A antibodies target A antigens, while anti-B antibodies target B antigens. If incompatible red blood cells enter the circulation, antibodies may attach to their antigens. The cells can clump together and break apart, causing a serious transfusion reaction. This is why correct matching is essential. The ABO system classifies blood using 2 surface markers, A and B, together with antibodies in the plasma. 4 combinations are possible: Group A, Group B, Group AB, and Group O. Group A red blood cells carry A antigens. The plasma normally contains anti-B antibodies, which can react against red cells carrying B antigens. In basic ABO matching, Group A receives A or O red cells. Group B red blood cells carry B antigens. The plasma normally contains anti-A antibodies, which can react against red cells carrying A antigens. In basic ABO matching, Group B receives B or O red cells. Group AB red blood cells carry both A and B antigens. Their plasma normally contains neither anti-A nor anti-B antibodies. For red cell transfusion, this allows an AB recipient to receive any ABO group. Group O red blood cells carry neither A nor B antigens. Their plasma normally contains both anti-A and anti-B antibodies. For red cell transfusion, O cells can cross ABO boundaries more easily than other groups. Here is the ABO pattern at a glance. A has A antigen and anti-B. B has B antigen and anti-A. AB has both antigens and neither antibody. O has neither antigen and both antibodies. The RH system adds another important marker, usually the RHD antigen. If RHD is present, the blood type is positive. If it is absent, the blood type is negative. RH status also affects red cell compatibility. Combining ABO and RHD creates 8 common blood types: a positive, a negative, B positive, B negative, AB positive, AB negative, O positive, and O negative. Other, less common blood group systems also exist. In the guide's approximate U.S. figures, O positive is 39% and A positive 30%. B positive and O negative are 9 each. The remaining types are less common, with AB negative near 1%. Red cell compatibility depends on which donor antigens meet which recipient antibodies. A compatible donation avoids markers the recipient would attack. UHD is checked alongside ABO, so the plus or minus sign must also be considered. For Rh positive recipients, A positive receives A or O red cells, B positive receives B or O positive receives O, and AB positive may receive any ABO type. Compatible cells may be Rh positive or negative. Rh negative recipients use Rh negative red cells. A negative receives A or O negative. B negative receives B or O negative. AB negative receives any negative. ABO group and O negative receives only O negative. In basic red cell matching, O negative is called the universal donor because it lacks AB and RHD antigens. AB positive is called the universal recipient because it can receive all 8 common red cell types. Plasma compatibility follows the reverse ABO pattern because the important factor is the donor's antibodies, not red cell antigens. AB plasma contains neither anti-A nor anti-B antibodies, making it the universal plasma donor. Universal does not mean testing is unnecessary. Before a planned transfusion, healthcare teams type the blood, screen for additional antibodies, and perform compatibility testing. ABO and RHD are essential, but they are not the only blood group systems.